to present my colleague, Dr. Ahmed Sallam, and let me please to introduce him in a few words. Dr. Ahmed Sallam is a senior researcher in the Desert Research Center, and he is currently working as postdoctoral researcher at the University of Illinois. Dr. Ahmed got his PhD in animal breeding and genetics from Alexandria University. Then he got 6-month scholarship to conduct research experiments in the University of Maryland, USA. He has rich experience in managing omics data such as phenomics, genomics, and the microbiomes. He also has a strong background in statistical genomics, quantitative genetics for complex traits, and genomic evaluation of livestock. Thank you, Dr. Ahmed, for showing up on time today to present your outstanding research. And please go ahead to start your presentation.
Okay. Thank you, Dr. Ahmed, for presenting me and giving me this opportunity to present some piece of work that you have been working on during the last 2 or 3 years. And I'm very glad to be here with you to present this, to share this information with you. So I really appreciate your invitation. Let us start with my presentation. So the title is quite different than the one that I have provided to you, because it was a while ago and some issues have been changed. So my title is quite wider than the other one. The title is Understanding the Inheritance of Complex Traits in Egyptian Goat Breeds. And I have only 2 outline. First, general introduction and background. My general introduction and background will be quite long, but it's very important to understand the context of the genetics of Egyptian goat and the project that we are working on. The second, which is genomic studies on Egyptian goats, which I will be presenting this information and presenting some results from our recent studies and some other studies that is still under investigation right now. So the story started many years ago, and when I'm talking about animal breeding and genetics. So I'm talking about genetic improvement of animals, which is mainly used by even selection or crossbreeding with other species. But it's mainly the genetic improvement is mainly based on first on selection. Even I'm going to do crossbreeding, I have to select the best animals first and then go for crossbreeding. So, and crossbreeding right now is a complex process, but it has a very long history that has been done many years ago. And the story started when a man from England called Sir Robert Beckwell, he has a farm and he has a very nice word, which is breed the best to the best. This is exactly what selection is doing. He was trying to evaluate his animals in the farm and then select the best animals based on the own information of these animals and the relatives, which we are called pedigree, and then started to use these animals and keep this animal in his farm to be used in as fathers or parents for the next generations. And then he had a very significant improvement in the production of these animals. And then the farms next to him started to do like him. And then they started to even share their selected animals with each other, which is known right now as crossbreeding. At the time, they didn't know that they are doing genetic improvement and animal breeding at a systematic way as we are doing right now. But this is the way that you can achieve and you can have more genetic improvement in your animals. But then, because the number of animals at the time was not that large, so they were able to keep tracking animals and identify the best animals without using any records. But by time things started to be more complicated, number of animals has expanded a lot, and more people were contributed to this process of sharing these animals. So it has been to develop a tool to be able to select these animals, to find the best animal among all other animals, to be kept and used as the fodder for the next generations. And just to imagine that if we have two parents, the one on the right and the one on the left, and I asked you which one is the best one to choose to keep in your farm, and it's apparently that you will use the one in your right hand, which is because it's heavier and most likely it has good genetic information that will we pass to the next generation, his next generation, which is his progeny and his offspring. So you will ensure that you will have good genetic information in your farm. But what if I provided you with another type of information, which is the feeding for these animals? So it seems also that the animal which is in your right hand has a very poor pasture compared to the other animal, which is the heavier one, which had a very good pasture to grow on. So because of that, we have to take into account all sorts of information when we evaluating our animals and calculating the best animals. We should include all sorts of information because you may, if you just neglect some source of information, something like weather or pasture, you may take the wrong decision and select the wrong animals which is heavier, and you think that it has a good genetics, but it is not. It is because of something else related to the environment. After that, the animal breeding and genetics as a science have been established of three main components. The first one, which is biology, and biology is related mainly to the phenotype or the trait that we are interested in, something like milk production or meat production. And you will be hearing this trait so many times during my presentation. So do not get boring of me. So we should understand the biology of producing this phenotype or this trait, because the biological context of milk production is completely different than the biological context of producing muscles, which in the end will produce heavier animals and higher carcass weight and higher meat production. It is completely different. The biology is completely different. The second is genetics. And then people, when they started to select for the best animals, which has the best trait, the best performance trait, they didn't know that this is the result of genetics. They know that there is something may inside the animal may contribute to producing this thing, but they didn't know that this is genetics. But and also the third component is statistics. So when you have all of this information, so many information have been added to the calculations, so many animals have been contributed. So it should be there is an approach or a tool to handle all this information, which is statistics. Apparently that there are three different stations have been influenced animal breeding and genetics than any other contributors. The first one is PCR invention and identification of the DNA as the genetic material of the mammals. So after that, people were able to isolate the genetic material and identify some candidate genes, and then integrate this information in the calculation or the calculation of the best animals, which is selection. Second is blob, which is best linear and biased prediction. So we are talking about prediction all the time. We are talking about predicting the future of the performance of our animals. So as much accuracy as we achieve in this prediction, as better as we want. So in blob, we were able to mix all the information that we have to do a better prediction with higher accuracy. And third, which is sequencing genomes, which started by sequencing the human genome, followed by sequencing the bovine genome, and then we were able to have a higher genomic information to be added to the calculations and the process of selection and genetic improvement. And this figure is just to show you that starting from 2009, which is after the completion of the bovine genome at 2007, there is a tremendously increase in the number of animals that have been genotyped and included in the genetic evaluation process in the United States only. And this is only until 2016. And I'm sure that the number of animals have been increased, maybe doubled this number. I know that maybe there is something like 5 million animals in their database. And this map is just to show you also the genotyped animals in the USDA database. And you can see that everything is color except for Africa. Unfortunately, it's still in black and white, and particularly in Egypt. And this is something we have to think in the future. We have to find a way to genotype our animals for genetic improvement or even for conservation of our genetic resources. Here I'm gonna present some results or output of this genetic information that has been going over so many years ago. We have noticed a lot in the increase in the meat production and milk production, and also the marketing rate of chicken after so many years of selecting the best animals, even based on pedigree or genomic information. And also using genetic information, we were able to identify single genes. And you may be aware of there are two types of genes, two types of traits. The first trait, which is more popular, which is called complex traits. And in this type of traits, something like milk production or meat production, so many genes are contributed to this phenotype, and each gene is contributed to this phenotype with a very small effect. But there are other types of traits, something like twinning or prolificacy in sheep or goat, which you may find one gene, or even double muscling in livestock. You may even find one gene which is called single gene will be able to make the effect of twinning or not or single birth. And also using this genetic information, we were able to identify lethal mutations. So this process of selecting the best animals, you may have a very good sire, which has a very good genetic information, and you wish to spread out his semen across your entire population. But this is very dangerous, because after so many times of using this sire and artificial insemination, most probably you will have something called inbreeding. So all bad genes will be accumulated in one sire or more, in one offspring or more, and this will brought up very serious mutations, which will lead at the end to losing this fetus or progeny. And without this genetic information, we were not able to identify these lethal mutations. And not last, this is one of the main issue of advantages of using genomic information, as we were able to do genomic evaluation. Then, which is we have animals which have been just born at 1 day birth, so we can, but we do not have any information about their phenotype. We don't know how much milk or growth performance this animal will be in the future. The only thing that we know is the pedigree, the relatives of this animal, and the genomic information. So we can collect blood samples, extract the DNA, and then scan their genomes, read the genomic information, and integrate this with other information, and we can have a prediction of the future performance of this animal with high accuracy after adding the genomic information to the process of calculations, which is called right now genomic evaluation or genomic selection. And lastly, a group of scientists, I think 3 or 4 years ago, were able to use this genomic information available after identifying the genes that are producing or the horn in cattle. And they were able to use the genome editing technique to produce samples without horn, and these horn are so harmful in the farm, and even it takes more space and may be harm or dangerous to the other animals. So this is a very good achievement. It's just a quick recap about the history of the genetic improvement and selection process. Starting before 1995, it was only based in pedigree and the animal's own information and other relatives' information. And in 2003, scientists were able to identify some markers and some restriction enzymes for those markers, which is called marker-assisted selection. And those were used as an assisted technique in the selection process. And after the completion of bovine genome in 2007, so Illumina was able to produce a SNP chip. Then you can send your DNA for your animals and read the genomic information about your animals. And the first version was 50K SNP chip, and then which is called the high density SNP that has more than 700,000 SNPs. And you can use this information to evaluate your animals, which will be included in further genomic evaluations. After that, the scientists all over the world decided to do whole genome sequencing for 1,000 ball, which is called 1,000 ball genome project, which are mainly used in artificial insemination. So we will have a lot of information about the genomes of these animals and their progeny, relatives, and offspring, and this also will be included or integrated in the genomic evaluation process. But the question about the future right now is, which is in a rise in the scientific media, do we have to sequence more animals to identify more new mutations to be integrated in the evaluation process? We don't know, because this is highly computational cost. So sometimes the 50K SNP chip is sufficient for our objective. So we don't know. We have to find some way of balance between the high computational cost and the benefits of sequencing all animals. Or do we need another source of variation between animals, something like epigenetics, which is controlling the gene function a lot than any other things? Or lastly, do more research on the microbiome, which is also a very good source of variation between animals, which is significantly contributed to the productivity of the animals. And lastly, we know some that the phenotype is a very big challenge, and some traits is hard to measure traits. So can we use computer vision and machine learning and artificial intelligence techniques to identify some novel accurate phenotypes and then use these phenotypes in the animal genetic improvement in the future? We don't know, but the future will tell us the answer of these questions. So a group of scientists in the Desert Research Center and I decided to apply for a grant to apply what we have learned outside in Egyptian goats. And we were lucky to be granted from the STDF in Egypt about 1 million Egyptian pound, which was at the time $50,000 for 2 years, to conduct some research using some advanced genetics techniques, which is genome-wide association analysis, in context of some important production traits in goats. And the reason of choosing goats, there is no specific reason, but first we are working in Desert Research Center, which is mainly focused on desert animals, and so we didn't have so many options to choose. We have just camels, small ruminants, which is sheep and goat, but we have choose goat because we thought at the time that there are so many variations between Egyptian goat breeds, and there are some types of production is more familiar in goats than in sheep, something like milk production and weaning rate. So we were interested in studying this type of production in goats than in other livestock species. The objective of this project was the first one was characterization of Egyptian goats based on their genomes, and the second one was to identify some genetic markers for important traits in Egyptian goats to be used in the future selection programs. The activities of the project was we just collected some phenotypes to do some genome association analysis, and we had a very large amount of phenotypes about milk production, pool efficacy, type traits, and feed efficiency. And the results of these GWAS gave us some positional candidate putatively potentially contributed to the phenotype of interest. So we were interested to go deeply in these candidate genes using PCR and single sequencing and some other techniques to identify some further mutations using SSCP and RFLP. And then for some studies, we used gene expression analysis for some genes using real-time PCR to see if there is a differential expressed gene between some traits or not. And we use the microbiome diversity and composition based on 16 srRNA. And lastly, we hope to use this information to calculate the genomic breeding values and then compare this with the breeding values based on pedigree only to see how much accuracy we got after adding this information to the calculation process. The breed that we have mainly was the Raibi, which is also known as Egyptian Nubian, and Berkey, and those are indigenous goat breeds. Berkey is located in desert, and Zoraibi is located mainly in the delta. And then we wanted to include some other imported animals, which was imported to improve our local breeds, which was, and we had access to Boer and Damascus goats. The Damascus goat was collected from one of our research facilities in Alexandria that belonged to Desert Research Center. And Boer were collected from a commercial farm in Delta, and Berkey was also collected from one of our research facilities in Oasis, in CU Oasis, and the Raibi was collected from one of these research facilities that belongs to Animal Production Research Institute in Delta as well. Our phenotypes, which was a big challenge for us, included body measurements, feed intake, milk production traits, twinning ability of each of these animals, microbiome diversity and composition, and pathogen infection. And we genotyped all animals in the project, which was, I believe it was something like 395 animals or so. We genotyped them using 60K SNP chip provided from Illumina, and this genotyping, all of the process of collecting phenotypes and DNA extraction and quantification was done in Desert Research Center. And then we shipped these DNA samples to FBN Germany for genotyping, as we have a collaboration right there. So for the objective one of the study, which of this research project, which was the characterization of Egyptian goat based on their genomes, we were able to publish our results in BMC Genomics. This map is to show us the locations of our collected samples. Here we collected Damascus breed from Alexandria and Matruh, and for Barki goat breed we collected from Siwa Oasis, and from a commercial farm in Delta we collected Bur, and from other places, which is Damita in north of Delta, we collected Zaraibi or Egyptian Nubian. The first thing we did is to plot PCA, principal component analysis, based on genomic information to see how close to each other our breeds. And we're quite surprising by this result, because we expected that Barki will be separated in a separate cluster than other breeds, because it seems that it's isolated in deserts. But we found that Barki is more related to Damascus and even Bur, and it was not a surprise to find Nubian and Bur was isolated in separate clusters, because those are breeds that are under breeding programs for so many years ago. So it's a kind of isolated than other breeds. But because I was the one who collected these samples, so I was asking the breeders about their breeding programs. So one of the breeders told me that the Barki goat breed is a very small in size breed. So to deal with this the small size breed, they were using one of the other large size breeds, something like Bur or Damascus, as a cross breeding with Barki to improve the size of this breed. So this may explain how close to each other, why these are close to each other, Barki and Damascus and Bur. So the main idea of this project to track the selection signatures, which are footprints that resulted from domestication and selection for specific important traits over so many years ago. And these traits are very important, something like immunity and reproduction and embryonic development and adaptation to the environmental conditions. So to achieve this goal, we used two different approaches. The first one was runs of homozygosity, and the second was FSCT, which is pairwise differentiation. The pairwise differentiation is the differences in allele frequency between different breeds, and it may give us a good indicator about the locations of the genome that are more under selection than others. And the runs of homozygosity are long stretches of, in the genome of a homozygote phenotype, which may a good indicator about that those animals share common ancestor, and may give us a better indication about the process of inbreeding. And all of this together are very important in the future breeding programs, which breeding programs that we should follow for our animals based on these results. So this is the runs of homozygosity for each breed, and this was not a surprise that we found that the long stretches, which was more than 16 megabases, was only found in Nubian breed, and this is because I said that this breed is under intensive breeding program that is being done in Animal Production Research Institute, which means that you will find accumulation of some genes, some genomic regions after this process of selection for a certain traits of interest, which is not the case for Barki, Bur, and Damascus. because those breeds were located in the boar was located in a commercial farm without any specific breeding program. And Barky and Damascus, they were new to our research facility, so they were not under any breeding program. While the very short segment of runs of homozygosity, less than 2 megabases, were the common in all breeds. And we also studied the inbreeding coefficient based on genomic information, and it also seemed that the Egyptian Nubian breed had the highly genomic inbreeding coefficient, followed by Boer, which is also not a surprise, while Berkey and Damascus had a very minor genomic information coefficient. And this information is very important when planning for future breeding and selection schemes in the future. And we took the percentage of the SNPs that appeared in all of the genome in more than 40% of the genome windows accounted for runs of homozygosity, which is called ROH hotspots. And this is an indicator for the genome, the regions in the genome that are under selection or domestication. And it seems that the region, there is a specific region in genome in chromosome 6 and chromosome 18. It was common in all breeds, which means that this may have something interesting in this. And after using the pairwise differentiation between breeds, it seems that the difference between Boer and Egyptian Nubian was the highest one, which is also not a surprise because this is an indigenous breed and this is an exotic breed. While the FST between the Egyptian breeds, including Damascus, which has been introduced to Egypt 50 years ago, is the minor ones. So we took this information and tried to investigate these genomic locations in chromosome 6 and 18 on the goat genome in the NCBI and in sample, and we find very important candidate genes, and all of them were related to adaptation, immune response, embryonic development, and reproduction, which makes sense to the goats that we are studying. We are talking about Barkey goat, which is predominant in desert. So it makes sense that it has accumulated genes for adaptation and immune response. And also we are talking about Damascus and Nubian, and those genes, those breeds are known as milking breeds and high prolific acid breeds. So it makes sense that we will find some interesting genes related to reproduction and embryonic development. This is just a quick recap about the first objective about genomic landscape of Egyptian goats. I just included here the difference between Egyptian Nubian and Damascus because they had the extreme values. So for Egyptian Nubian, the highest mean accumulative ROH, which was 360 megabases in length, compared to 40 megabases in Damascus. And this is, as I said, because the Egyptian Nubians are followed by a strong breeding program in APRI. And also, the Egyptian Nubian exclusively had the longest ROH segment compared to Damascus that had, or even the other old breeds. The less than 2 megabases were common in all breeds. And also the genomic breeding coefficient was the highest in Nubian, followed by Boer, compared to the lowest values in Damascus and Barkey. We identified a very high signal in chromosome 6 and chromosome 18 based on FST and ROH hotspots. And these genomic regions included important genes for immunity, reproduction, and adaptation. The second objective of our research project was to identify genetic markers for important traits in Egyptian goats. and we were able to publish the results of this objective in scientific reports. And this had a story. We didn't plan to have this activity in our research project, but because we were interested in collecting milk samples from animals, so we were planning to track these animals for the period of milking, to collect milking samples and to calculate the milk production for each animal. But one of the farms that we were following, and all of them were pregnant, and the total number of them was 76. Suddenly, at the third part of pregnancy, half of them were aborted. And this is really shocked us, because it means that we lose 50% of the animals in this farm. And you know that this phenotype is a big challenge for us. But we took this as an advantage for us, because it seems that there is something in the genetics, because those animals were nearly at the same age, and they eating the same diet, and they are in the same place, and they are in the same pregnancy stage. So what has happened that made those animals to be aborted, while the other animals keep pregnancy and gave a normal birth? So we asked one of our research contributors, who was a veterinarian, and he suspected that this may be brucella infection. So we took the protocol to identify this brucella infection using ELISA and Ross-Bengal test, and we did this twice or 3 times to confirm the phenotype, because this is very dangerous when you run genome-wide association analysis for diseases or susceptibility to disease infection. You have to make sure that your phenotype is accurate and correct. Then we genotyped these animals using the 60K SNP chip, and we conducted the genome-wide association analysis, and we were very lucky to find a very good signal for some candidate genes that may be contributed to the susceptibility or tolerance to this dangerous disease. And also we collected fecal and vaginal swab samples to see if the infection status of brucella has affected the diversity and composition of the microbiota or not. And we did this based on the 16S DNA gene using paired in Illumina sequencing. Results of our study, we used 3 different approaches for GWAS. The first one was FST, which is pairwise differentiation based on allele frequency. The second one was single SNP at a time, which is a mostly common one. And the third one was genetic variants explained by windows. So we divided the genome into windows, and we asked to find the leakages equilibrium between those SNPs in each window. And we identify the genetic variants explained by each window, and the windows that explained more variance between individuals, it means that it has something interesting in it. And we only consider the genetic markers overlapping between all of these 3 approaches as a strong, significant signal for something interesting in it, which was on chromosome 15. And when we looked at the putative candidate genes that may be in chromosome 15 at this region may be contributed to susceptibility to diseases, we found a very interesting gene which is involved in pathways related to TLR and interleukin receptors pathways, which makes sense for our study. And when we came to the differentiation, it is clear that based on the 16S rDNA in the microbiota, it is separated the infected and uninfected animals into two separate clusters based on vaginal swab and fecal swab. And it was also clear that the microbial diversity increased in the infected animals compared to the non-infected animals, and also infected animals showed a higher relative abundance of the pathogen bacteria compared to the non-infected animals. And the most important thing that this difference in the composition and diversity of the microbiota has affected the ability of animal to the rumen digestion and feed efficiency and absorption of the nutrients. And this thing should be taken into account when making diet to the diseased animals. And we talk because we had a very good result from this one. We just took 15 genes and try to find if there is these are differential expression genes between infected and uninfected animals, and we had a very good signals between differentially expressed the genes between infected and non-infected animals with Brucella, and this information is not published yet. And also, we are still working in this right now, but we had a very good signals for milk traits and Damascus in Damascus goat breed, and also in Barki goat breed. And I'm just presenting this information because people in Egypt think that they may think that our local breeds are not good enough, but no, science tells us that they are good. We have very good signals based on Manhattan blot and genomic information, and there is no bias in this, by the way. And also for the morphological traits, we nearly have single genes, as you can see this signal here. And based on QQ plot, it seems that our result is correct because the observed and expected SNPs, each dot of this is the marker, are matching together, except for some very few SNPs which are significant for the studied trait. The conclusion, based on the results of our study, we can conclude that multiple selection signatures were detected in 4 goat breeds in Egypt using ROH and FST. We identified chromosomal regions as a very interesting candidate genes for immunity, reproduction, and adaptation. And also we identified novel genetic markers for susceptibility to brucella in Egyptian goats. And we were able to identify that brucella infection influenced the microbial community and diversity. And this thing should be taken into account when selecting the diet for the diseased animals. We were able to identify 50 genes that were differentially expressed between infected and non-infected animals with brucella. And this information could be used as genetic markers for susceptibility to the disease in goats in the future. And here I would like to acknowledge the contribution of so many people has been contributed to this work, as you may see. But I would like to gratefully thank our colleague from Desert Research Center, Dr. Ibrahim Abu Sliman, Dr. Ahmed El Shinrawi, Dr. Ala Rabia, and from our collaborators from Animal Production Research Institute, Dr. Adel Abu Naga, Dr. Shaima Muhammad, and from Dr. Hamdi Abdel Shafir from Cairo University, and Dr. Camila Braz from the University of Illinois at Urbana-Champaign, Dr. Klaus Wimmer and Dr. Henry Reyer from FBN in Germany, and Dr. Francesca from the University of Bologna in Italy. And thank you all again for giving me this opportunity, and I'm ready to take any question. Thank you.
Thank you, Dr. Ahmed, for really very interesting lecture and we got very useful information about the Egyptian population of goat. And now we open discussion for this lecture. So please, if you have questions, you can write your question in the chat room, or just you raise your hand and I will unmute your mic.
Thank you.
Any questions? So I may start, Dr. Ahmed, for the first one. Regarding to the good population you collected for your genomics studies, I can see that you didn't cover good individuals from different part in Upper Egypt.
Yeah.
So I think why you didn't do that.
Yeah, actually, as I said, the phenotype, it's very easy to collect DNA sample from any individual, and just you can read the genomic information using the Illumina scanner. It's very easy, even if it's costly, but it's easy. But the big challenge in our genome study, and you may know that, Dr. Ahmed, and even it's more challenging in our field in animals, that you find the good phenotype to follow. So we were interested. It was a big challenge for us to collect phenotypes and because we had to follow for something like milking production, we we had to follow the animals for like 4 months or 6 months. Each month I had to go to the farm twice to collect milk sample and take this sample directly to go to the lab in the same day to identify the milk composition. So it was very hard to us to go very far from our research facilities that we had labs in it to be able to do this analysis. But of course, we in the future research project we may include the Upper Egypt in that if we find collaborators that may help us to do this analysis.
Okay. And about the SNP marker that you did, how homozygosity you found in the population? I mean, do you find high homozygous SNPs, or the most of the SNPs were with heterozygous loci?
Yeah, actually, thank you for this question. Tag. It is a very good question, and it actually based on the breed. For example, the breeds that are under specific breeding programs by research institutes or universities, so they are taking care of them, such as Nubian and Boer. Yeah, number of homozygous SNP was higher than other breeds, which is Berkey and Damascus. And those Berkey and Damascus, they were brought to our research institute at the same time when we started the project. So we didn't find a large number of homozygosity on them. And this
And
is, yeah, we were lucky to have this result because this was a very good source
yes.
of variation for us to study on it.
So you did your GWAS with heterozygous loci. You used the heterozygous loci in the GWAS analysis as they are, because in blend breeding, so we we removed mostly in wheat, for example, we removed the heterozygous loci from the marker data set, and we prefer to conduct GWAS with only homozygous loci.
No, I don't think so. You know that there is a step at the beginning of the GWAS, which is called quality control filtration process. And we just removing animals or SNP that didn't have enough information to be included in further analysis. But we include all SNPs, including those are heterozygote and homozygous SNP, and even those not in high Weinberg equilibrium, because high Weinberg equilibrium is one of the results of this evolution or selection. So this may be we may remove SNPs that are important for us. So it depends on the objective of your study. Yeah.
Okay. We have a question from Ibrahim. Yes,
Okay.
I unmute your mic. Please go ahead.
Hello, can you hear me?
Yes.
Yeah, I hear you.
Yeah, okay. First, I'd like to thank Dr. Ahmed Sallam for his interesting presentation about goat breeds and its genomics in Egypt.
Thank you.
The second thing, I want to clarify your question, Dr. Ahmed Sallam, about the Upper Egypt breeds. So in Upper Egypt, the main breed is the Nubian breed, as Dr. Ahmed Sallam mentioned in the first of presentation. And we already collected samples from Nubian goats, but from different places. So we already covered the Upper Egypt area in our study, depending on collecting samples from Nubian breed. This is, I just want to clarify this point.
Okay, thanks. Yes. Thank you, Dr. Ibrahim. Thank you.
Thank you.
Do you have question? Yeah, we have a question in the chat, Dr. Ahmed. I saw a picture of an animal that has double muscles. Is the meat of this animal harmful to humans when eaten? Does the animal have any behavior such as aggression?
This is a very good question. I do not have enough information about this topic, but in terms of harm of all of this meat to for human, I don't think so, because most in the abroad we have two different lines of cattle, which is beef cattle and dairy cattle, and they are completely different in terms of everything, genetics, behavior, and everything. So and most of animals here in the US, for example, in beef cattle is like this. They are high masculinity compared to dairy cattle, which is very small with big udder. And in terms of behavior, yes, beef cattle is more aggressive than dairy cattle, which means that it is really hard to collect phenotypes from them. Right now in the US, they are using, as I told you in my introduction, they are the lab that I'm affiliated right now. They are using computer vision and sensors integrated with machine learning and artificial intelligence to collect phenotypes from those aggressive animals. I'm not sure if this answers your question or not.
Okay, thank you, Dr. Ibrahim. Any other question? So I think we come to the end of the presentation. Once again, I would like to thank you, Dr. Ahmed, for the nice presentation. And we will meet again next week in the Dr. Hiwan from Ethiopia. We have a presentation on SNP marker diversity in avocado. So I hope that you attend this lecture next week. Thank you for all,
Yeah.
and thank you, Dr. Ahmed, so much. Thank you, and we hope
Thank
to
you.
see you again with
It was my pleasure.
useful information next semester, inshallah.
Sure. I will be glad to share this information with you. Okay.
Okay. Thank you. Thank you.
Thank you. Bye.
Bye. To all attendees, we will leave the link of registration for like 15 minutes. So anyone missed the registration can now register for the 10 minutes. Thank you and see you next week.